Hello everyone, welcome to the Knowledge Hub by Sri Chaitanya. This is Sayyid Danish Anjum, Senior Zoology Faculty, and in today's session we are going to study about some of the basic concepts of the phylum Porifera. So starting with the basic concepts of the Porifera, without a delay, let's start with the classification of the kingdom Animalia. Now Porifera, as we know, it's one of the most primitive phylum in the kingdom Animalia, right? So if you see the classification of kingdom Animalia, we see that Animalia or Metazoa, kingdom Animalia, just a moment please. Right. So if we see the classification, whole of the animal kingdom is classified into two parts. Now the first is the subkingdom Parazoa and the next one is the subkingdom Eumetazoa. Eu means true and Metazoa means animals. So in the kingdom Parazoa there is no differentiation, there are no tissues, there are no germ layers, and hence it is the most primitive and the basic classification of the kingdom Animalia. That means subkingdom Parazoa will not be having any higher level forms like tissue level organization or organ system level of organization. So it will be having the most basic concept of level of organization, which is cellular aggregate level of organization. Now if we see that only phylum Porifera comes into this sub-kingdom Parazoa. Apart from that, all of the other examples of all of the other phylums are categorizing into sub-kingdom Eumetazoa. Now, further, on the basis of this symmetry, right, as you have studied in the previous sessions, that the symmetry is of two kinds, bilateral symmetry as well as radial symmetry. Also we have studied coelom as schizocoelom and enterocoelom, and on the basis of the blastopore development we have studied that the organisms are categorized either into the protostomiates or deuterostomiates. So what basics we studied in the previous sessions now comes into the classification. So we can see that grade radiata include particularly those organisms which are having radial symmetry, which is the first point. Second, on the basis of the germ layer organization, they are categorized as diploblastic. Now we have studied in the previous session that on the basis of the germ layer organization, the organism can be categorized either into the diploblastic or into the triploblastic. So in the diploblastic, the organism will be having two germ layer, that is ectoderm and endoderm. Mesoderm will not be present. Second is the, as you can see, the grade is the bilateria. Bilateria means, or bilateral symmetry organism means that in only one plane we are getting two identical half. So these organisms are usually triploblastic. So in these organisms, three germ layers will be present: ectoderm, endoderm, and then the mesoderm, which is the middle layer. And further, this category is classified into two further categories based on the blastopore development. So if we take the blastopore development, then if blastopore opening develops into mouth first, then such kind of organism will be categorized in protostomiates, or if it is developing into anus first, mouth later, then it will be categorized into deuterostomiates, right? So these are the two categories, protostomiates as well as deuterostomiates. So these are the superphylum. Now, as we can see, on the basis of the coelom development, we have studied in the previous session that in the coelom development, organism can be categorized into three categories: acoelomate, pseudocoelomate as well as coelomate, right? So these are the three categories on the basis of the coelom cavity. Now as I have defined the coelom as the cavity which is enclosed by the mesoderm germ layer, so definitely in the diploblastic, coelom will be not present. Even if the cavity is present, it will not be termed as coelom cavity, right? So if you see that protostomiates, or those organisms in which blastopore is forming mouth first, these are categorized into pseudocoelomate. Now pseudocoelomate, only one phylum comes into this category, which is Ascarianthes or Nemathelminthes, or we can call it as Nematoda, which we are going to study further in the upcoming sessions. Further, the other two categories, which are Acoelomate and Eucoelomate or Trucoelomate, are categorized into the category Bilateria or Triploblastic bilateral symmetry organism. Now we see that in Acoelomate, no body cavity is present. So one of the example of triploblastic Acoelomate is Platyhelminthes. Mark my word, I have said triploblastic Acoelomate. Diploblastic will also be categorized into Acoelomate. Even Porifera in the NCERT is categorized into Acoelomate, right? So you have to know the difference between triploblastic and diploblastic Acoelomates. Fine. As we see, pseudocoelom or pseudocoelomates, only one phylum is there, that is phylum Nematoda, and Rotifera is the another phylum which has not mentioned into the classification chapter. Its introduction has been given into the excretory chapter, but in detail Rotifera is not mentioned. So as of now, we can say that according to the syllabus, phylum Nematoda or Nemathelminthes comes into the pseudocoelomate category. Now in the coelom category also, we have studied in the previous session that coelom also develops in the two types, schizocoelom and pterocoelom. Schizocoelom by mesodermal splitting method, enterocoelom by gut pouch method, right? In both of the methods, the formation of the coelom is different. That means the approach to form the coelom is different, but at the end coelom cavity is forming by mesodermal lining only, both in schizocoelom as well as in enterocoelom. So if we see that in schizocoelomate, coelom formed by splitting of mesodermal mass, and in this method the phylum examples are Annelida, Arthropoda, Mollusca, right? So in this method we have seen in the previous session also that the mesodermal mass develops from both the sides and finally it combines and form a complete enclosed cavity which we call it as coelom, correct? In the enterocoelom, we know that coelom is formed by pouching of archenteron cavity, right? So example of these enterocoelomates are phyla Echinodermata, Hemichordata and Chordata. So out of these schizocoelom and enterocoelom are considered more coelomate, that means more true coelomates as compared to schizocoelom, but both are coelomates only. Fine. So this was the brief category of the classification of animal kingdom. Starting with the Porifera, let's study its basic concept first. So as we see that during evolution, the first life form was unicellular in nature. Later on, this unicellular habit changes towards the multicellular form. So in between the unicellular form and the multicellular form comes a connecting link which is called as Proterospongia. So if you look at the cell of the Proterospongia, it is a unicellular eukaryote and it evolved from coenoflagellate. That means Proterospongia is a coenoflagellate protozoan. It is a unicellular eukaryote, and Proterospongia is the connecting link between protozoa and Porifera. Why? In the earlier session, we have studied that the Porifera is a cellular aggregate, correct? And all the cells, although behave independent in the manner, but they are having some sort of coordination and some sort of division of labor among them. But as we see in the case of Proterospongia, you can see in this diagram that the cells are totally independent, but they live in a group or in a colonial form. So in this way, they behave as a multicellular form, although they are not strictly multicellular in nature. So this we have to keep in mind. That is why we can say that from this Proterospongia, from these unicellular coenoflagellated protozoans, the multicellular level life forms begin to come into picture or begin to come into existence during the course of evolution, right? So you must keep in mind that Proterospongia are the connecting link between the unicellular form and multicellular form. That means between Protozoa, which is the unicellular form, and Porifera, which is the multicellular form, correct? So you can see here that the root or the tree of the classification starts with the protozoa, and between the protozoa and the eumetazoa, that means the complete U means true, there comes the parazoa. We can also classify this also, sometimes we can interchangeably use for it as a connecting link, but strictly speaking, connecting link will be Proterospongia. Fine. So eumetazoa on the basis of symmetry further classified into two parts. Radiata means radial symmetry, bilateria means bilateral symmetry. Further, they are also categorized into further protostomia and deuterostomia. That means on the basis of the blastopore development, and further on the coelom basis, acoelomate, schizocilomate, and further we can see the other examples like chordata, echinodermata, and schizocilomate mollusca, arthropoda, annelida. Similarly, acoelomate, triploblastic acoelomate, platyhelminthes. In this category, diploblastic will also be categorized in acoelomate only. So we can also categorize Cnidaria as well as Ctenophora in acoelomate also. Even Porifera in NCERT is categorized in acoelomate. Okay. So the subkingdom Parazoa we are going to study now, and the study of sponges is known as parazoology, right? So what are sponges? Basically, we have studied in the very first basic unit of classification that the cellular aggregate form or cellular level of organization is the basis of the formation of Porifera. Now Porifera are pore bearer. In the common language, in the common name, we term them as pore bearer or sponges. Here we can see that they are also known as sponges. So member of this phylum are commonly known as sponges. Now these are, as we have talked about, that during the course of evolution these come first after the Proterospongia, when the multicellular life form was evolving. So the first true, or the after just the Proterospongia, the first multicellular life form which comes into the existence was the Porifera. So these are the most primitive multicellular animals and have cellular level of organization. So here we can see that we have taken an analogy of the sponge, that as in sponge there are numerous tiny pores present in the body which forms a system, which forms a network inside the body of a sponge. Similarly, in the body of the Porifera, we have many numerous pores or ostia. You can call it as pores or ostia, which are present on the surface of the body, and they form a complex network, so by which the water can enter and forms a water canal system, by which they can have their nutrition and they can have exchange of nutrient as well as waste. Fine. So if we see further that the habitat involves aquatic, that means they are not terrestrial. So totally, exclusively the habitat is aquatic. But when we call it as aquatic, we have to further classify that too into mostly marine, and some examples are of freshwater, that is spongilla. So the common name of spongilla is the freshwater sponge, and most of the other Porifera comes into the category of marine habitat. Okay, so you have to remember this exception. Here we can see that this is the marine example and this is the spongilla, that is the freshwater sponge example. Now, as we move further, we can see that the habitat of these organisms is mostly sessile in nature. Now, students, what is the meaning of sessile? That means whenever an organism through its body part gets attached to a solid substratum, either it's a solid rock, either it's a floor. Whenever an organism through its body part gets adhered or attached to a solid substratum, solid rock, something like that, then such organism is said to be sessile in nature. That means whenever the organism is sessile, it will get stick to its one place, it will not move from its place, it will not show locomotion. Whereas in the free living or free swimming, you can see that the larva, which is the free swimming stage, it freely swims due to the presence of the flagellated cells, and when it finds a suitable substratum, it gets attached to that and it develops into the adult organism. So that's how we can see that there are two habitat. Mostly the adult are sessile and the larvae are free floating or freely swimming. Okay, now as you can see that mostly they are asymmetrical in nature. That means they do not have a regular shape, but however these asymmetrically nature is also having an exception. For example, Leucosolania and Sycon. So these two are examples. I will write it down, and also these examples are coming further also. So these two are having radial symmetry, but apart from these two, mostly Porifera are irregular in shape, and therefore they do not have either bilateral or radial symmetry, so they will be asymmetrical in nature. As you can see that there is no symmetry we can guess into such kind of organism which are having a irregular shape. Fine. So moving on further to the level of organization, we have mentioned numerous times, multiple times, that these are having cellular level of organization, and the cellular level of organization is having a loose aggregated group of cells in which each cell behave more or less in an independent manner, but they are having some sort of coordination among themselves and some sort of division of labor among themselves. Fine. So here we can see, level of organization, cellular level, and cell aggregate body plan is there. Okay, here we can see in the picture, in the diagram, that this is a schematic representation of the phylum Porifera body, where we can see that this is the cavity part, this is the pore through which water will enter. Here the water canal system pathway is also shown, which we will discuss further. So there are two layers we can see. The first layer is towards the cavity, which I have marked with this. The second layer is towards the outer side, which is also called as the dermal layer. So we can call it as dermal layer, which is outside, forming the outermost layer, and the innermost layer is the gastral layer, which is towards the cavity. Fine. So we are going to study further how these layers are made. So level of organization is cellular level of organization and cell aggregated body plan. Loose aggregation of cell, we sometimes use this term also, in which the cells have little coordination and little division of labor present among themselves. Each cell behaves in an independent manner. Now, as we move further, that they are also having endoskeletal support, but as we can see that in our case we are having bones for the endoskeletal support. These are not having any proper endoskeletal supportive structure. Instead, their endoskeletal support comes from two major substances. The first one are the spicules, which we can see that are of various shapes like this ones, and the other part is the spongin fiber, which is nothing but it is the proteinaceous fiber like the collagen fiber, which forms a network and so provides rigidity and strength, and hence forms a part of endoskeleton. So here we can see that skeleton has two major parts, spicules. Now the spicules, which have different shapes, as we can see that, for example, triaxon has three rays which are needle-like shape. Diactinal has two pointed ends. Tetraxon has four pointed ends. Monoaxon amphidisc has dual side umbrella shaped pointed ends, and hexaactinal triaxon has 6 rays. Spongin fibers, as you can see, it's a network of fibers. Now, spicules on the basis of the composition can be categorized into two parts. Further, the first, those who are made up of calcium carbonate, they will be called as calcareous spicules, and those who are made up of silica, they will be called as siliceous spicules. So I'll write it down that those who are made of calcareous Those who are made up of calcium carbonate will be called as calcareous, and those who are made up of silica, they will be called as siliceous. Like this. Okay. So the body of the Porifera will be having either of the spicule, either of the calcareous or either of the siliceous spicules. So on the basis of the spicules also, the Porifera are divided into different classes. So the other part is the fiber, which is the none other than spongin fibers. But there are some exceptions to it, that some lack skeleton, example Oscarella. So this is the only exception which lacks any endoskeletal structure. Now moving to further, that the body wall of these organisms, Porifera, can be divided into two. The outermost part, dermal layer, the innermost part, gastral layer. Now if we see the diagram of the Porifera, like in this previous one, here we can see that the body form has two layers, the outermost the dermal layer and the innermost the gastral layer. In between these two layers, we have a non-cellular layer which we call it as mesohyl or mesenchyme also. So the third layer which we can see is the mesohyl or it is the mesenchyme. Now this you must remember that it is a non-cellular layer and these two are cellular layer. So overall we have three layers, two cellular and the middle one is the non-cellular layer. In this middle layer, mesenchyme, we have different types of cells also, which we call it as amoebocytes. So there are many types of amoebocytes having different kinds of function. We will see what type of amoebocytes we are having, and we can see that in the inner part of this body, we can see that there is a cavity present. This cavity is called as spongocoel or paragastric cavity. So this particular cavity will be called as spongocoel or paragastric cavity. Right, what happened? Cavity. Right. So paragastric cavity. My mark is not working somehow. Okay. What happened? Right. So paragastric cavity. Let me mark it again. Okay. Fine. So this is the cavity part. This is the outermost layer. So overall we are having three layers in the body, right? So we will see that dermal part consists of which layer of cells and the gastral part consists of which layer of cells. So here we can see that the outermost layer, dermal layer, that means the body wall, here we can see the body wall is distinguished into the outermost pinacoderm layer, which is also called as the dermal layer. It is the outermost layer and consists of flattened cells known as pinacocytes. So these are the flattened cells which are called as pinacocytes. And in between you can see that there are oval shaped cells known as porocytes, which forms the pore known as ostia. So here we can see that these are the porocytes which are forming the pore, ostium, plural we can call it as ostia, and the outermost layer is made up of pinacocytes, and the innermost layer is made up of choanocytes. So innermost layer choanoderm made up of choanocytes, and outermost layer pinacoderm made up of pinacocytes. Now as we look at the inside layer, this choanoderm layer, which is made up of choanocyte, it is the most important sole characteristic of the phylum Porifera. So choanocytes or collar cells, these are the basic characteristic cell of the phylum Porifera. Fine. Now in between these two layers we can see that the third non-cellular layer is the mesohyl. It is a middle layer, non-cellular, and it is a jelly-like layer which is having many other cells also, as well as it is the layer which is having spicules as well as spongin fiber for the endoskeletal support. So if somebody asks you that which layer provides endoskeletal support in the phylum Porifera, it will be mesohyl or mesenchyme. Fine. So here is the arrangement of all the layers. We can see that these are the spicules, particular, and these are the different other type of cells which we call it as amoebocytes, and this is the jelly-like layer, composed of jelly-like substances, and in between the spongin fibers will be also present. Okay, so moving further, we see that the outermost layer, pinacoderm, outer layer of body wall, contains pinacocytes and porocytes. Here we can see in the diagram. Then the inner layer of the body wall is called as choanocytes. Inner layer is called as choanoderm, and it is made up of choanocytes or collar cells. Now choanocytes line the spongiocele and the canals. Right? So it is the characteristic cell of the phylum Porifera. It is a flagellated cell. Now choanocytes, we can see that its shape, if you see that it has a shape of a collar. So that's why the common name comes collar cells, and it is also the characteristic cell, and also it has the property to distinguish into or differentiate or give rise to germ cells also. It also do the process of ingestion. That means food particle will comes into the contact of the choanocytes, and the choanocyte will ingest the food particle intracellularly. The digestion will happen intracellularly, and in this way each and every cells जो है it takes its own food material and it behaves independently, right? So what is the role of the flagella? We will see its prominent role in the water canal system shortly. So choanocyte line the spongiocele and the canal, or it lines the spongiocele, as well as we can call it as a paragastric cavity. So we can see that in this case there is no particular digestive system, no particular respiratory system, no particular circulatory or excretory system. So when it comes down to the point of digestion, digestion will not occur in the gastric cavity. Digestion will not occur in this gastric cavity spongiocele. It will occur only and only inside the cell. Hence the digestion in this case, in this phylum Porifera, is intracellular only. Okay, so now apart from these two cells, pinacocytes and choanocytes, the other type of cells which are having different functions and collectively they can be categorized as amoebocytes because of their shape. We can categorize them as sclerocytes, producing spicules. So if they are producing calcareous, they will be called as calcoblasts. If they are producing siliceous spicules, they will be called as silicoblast. Now spongiocytes, that is producing spongin fibers, which are the fibers responsible for endoskeletal support and provides rigidity. Archaeocyte, now this is one of the most important type of amoebocyte. Why this is the most important? Because it is totipotent in nature. Totipotent cells are those type of cells which can differentiate and give rise to any other type of cells of the body. So these are the type of stem cells that you might have heard. Similarly, these are totipotent cells which can give rise to any other type of cells, whether it is pinacocytes, whether it is choanocytes, or whether it is any other type of amoebocytes, right? The other type of cell is the trophocyte. This is the nurse cells which provide food to developing cells. Colonocytes secrete collagen fibers and forms endoskeleton. Now collagen fiber is a fibrous protein which provides strength and rigidity and provides mechanical support. And you must also remember that collagen fiber, as we have in our body also, it's not the same. Collagen fibers are of different type and different classes also, type 1, type 2, type 3, so on. So apart from that, colonocytes are in general having a role to secrete collagen fibers. Myocytes. Now the myocytes are those particular cells which lines the osculum opening, and in this way, as the name resembles with the muscles, they have the ability to close or to open the osculum opening. Now where is the osculum opening? Let us see in this figure. This one. This particular opening is called as osculum. Okay, right? So these cells control the opening and closing of the osculum. Okay, now what about the body cavity? As I have mentioned, spongiocele or paragastric cavity, it's one and the same thing. And here we can see this is the particular cavity, this one, in which the water will enter and it will leave through the osculum part. It's a part of the, It is a part of the water canal system. Now, before moving further, I want to let you know that you can register with us through scanning this QR code, and you can learn more about our courses and more about our session. So, okay. So, moving next to important property of this phylum is the water canal system. So now, if you see the water canal system, you might get confused with a water vascular system, which we are going to study in echinodermata, not right now. So, water canal system is particularly a network of the canals in which the water enters and leaves the body of the porifera. Now, what is the main use of the water canal system? By moving the water inside the body or inside the cavity, through the water comes the food, and the food particle, when it comes into the contact of the choanocytes, they ingest it, they phagocytosis them, and in this way the digestion occurs, right? So water canal system also helps in respiration, that is gases exchange, and also in the transportation of waste material. Fine. So these are the benefits or advantages of the functions of the water canal system. Now let's take a look. A non-toxic yellow dye has been squirted around the base of a purple tube sponge in the Caribbean. That means the scientists there, what they did is they injected a fluorescent dye nearby the body of the porifera, and in few minutes only we see that this dye was getting absorbed into the body, into the surface of the porifera, and it was emitted out in a squirting manner through the osculum opening. So you can see that this depicts that the flow or the pathway of the water canal system. Shortly thereafter, the dye is pumped out through the osculum at the top of the sponge. So what does this show? That the water enters through the body surface. Now we have understood, we have taken a look through the body surface that numerous tiny pores are present, which we call them as ostium. So through ostia, water will enter and it will come into the spongiocele or the paragastric cavity, and through the osculum it will emit out. Okay, so this is a normal pathway. So I want to let you know that on the basis of the water canal system also, the porifera are categorized into the three categories: ascon type, leucon type, cycon type. So out of these three categories, as we move further, ascon type is the most simplest type and leucon type is the most complex type. So the water canal system gets much more complex, gets much more branched, and in this way there are other canal system, but the whole pathway almost remains the same, in which the water enters through the ostia, and in the higher system the enters into the various canals, ultimately coming to the spongiocele or paragastric cavity, and ultimately leaving through the osculum or the opening. Okay, so this is the water canal system. Now, as you can see, sponges have a water transporter canal system. Water enters through minute pores called as ostia in the body, and it comes to the central cavity spongiocele, from where it goes out through osculum. That is the outermost opening. Ostia inhalent apertures, that means the water is ingressing, water enters through this pore, so we have termed it as inhalent apertures, and osculum, through which the water exit, we can call it as exhalent aperture. So here we can see the normal pathway: ostium, through ostium, this is the pore, it enters into the spongiocele, this is the central cavity, and through osculum it is leaving. Okay, now functions of canal system. As we have mentioned earlier, food gathering. That means when the water will enters into the body, it will come along with its food material, food debris. So then those food particles, when it comes into the contact of the choanocytes, they will phagocytosis, and hence the intracellular digestion will occur. So in this way, the water canal system helps in food gathering. Respiratory exchange, simple it is, that the water has dissolved gases, so the oxygen will be directly exchanged to the cells and carbon dioxide will be directly exchanged with the water. Fine. So respiratory exchange and removal of waste. So here the nitrogenous waste will be directly expelled out from the cell to the outer environment, which is water, and also helps in sexual reproduction, and in that too in cross fertilization. We will see that how this cross fertilization happens in the reproduction part. Okay, so one thing important you need to know, that if the water canal system is in such a way designed that water enters through the ostium pores only, and it enters through the spongiocele and leaves by osculum, one might get confused that water can enter through osculum also and the reverse pathway can also happen. But to ensure that the pathway is in a single direction only, the flagella of the choanocytes, they beat in such a direction that water always flow into this particular direction, in particular one direction, which is towards the osculum, and in this way it ensures, the beating of the flagella ensures that water will enter and it will flow and its movement will be continued towards one direction only. Okay, now nutrition, holozoic mode of nutrition, not to mention. So we can see that they are commonly called as filter feeder. Now what does filter feeders means? That whatever the food material present in the water, they will separate it from the water and they will have it as a nutrition. So suspension feeders or filter feeders, they are called as microphagas. Digestion is totally intracellular and it occurs in choanocytes and trophocytes. Right? Now moving further, we can see that this diagram depicts that when the organic substances, the food material along with the inorganic substances enters through pore, archaeocytes, choanocytes, engulf it, carry on the digestion, and the rest part leaves by the osculum. Okay? So, gases exchange, respiration and excretion, diffusion through general body surface, they are ammoniotalic. That means the nitrogenous excretory waste will be ammonia. Okay? And now coming back to the coordination, there is no nervous system of any type. So nerve cells and sensory cells are absent. Pinacocytes or certain myocytes are contractile cells that can respond to environment by decreasing surface area or closing osculum. So we have seen that in the other type of amoebocytes, we have seen that there were cells called as myocytes which were regulating the opening and closing of the osculum. Okay, so now the important part is the reproduction, upon which many questions comes regarding to different phyla, and this is the main part where student usually gets confused. Now if you see the reproduction method, the phylum Porifera reproduce both asexually as well as sexually. Now, sponges reproduces asexually by fragmentation and sexually by formation of gametes. Now, here one more point I would like to add, that sponges also have a great regeneration power. So, when I say regeneration power, that means more or less I am talking about the fragmentation also. So, in a common sense, fragmentation as well as regeneration are more often used as interchangeably terms, but there is a minor difference. But when we say that an organism is reproducing asexually by fragmentation, that means it is to be understood that the organism is having very high regeneration power. Then and then only it can form the rest body part from a fragment. Okay, so they can reproduce both asexually and sexually. Now, asexual method involves multiple types. For example, fragmentation, which I have already just explained to you. Apart from fragmentation, we have other two methods. The first is the budding. Now the budding can be further distinguished into the internal budding as well as external budding. So for example, if there is bird which is arising on the surface of the body, that birds get detached and give rise to another offspring, this type of budding will be called as external budding. And if the budding happens in the internal cavity or inside the body of the organism, then such type of budding is called as internal budding. So internal budding has a specific name for it, gamules, while we can see that in a normal budding method, the fragmentation as well as external budding helps in the asexual reproduction of phylum Porifera. So we can see that when the bud gets detached or if it gets fragmented into various species, each fragment has the capacity when it gets attached to a solid substratum, it will give rise to a new organism. So they will be all clone, and the offspring will be clone or exactly genetically similar to the parent, as in the case of asexual reproduction. Now, as we see in the budding, it is an exogenous budding, that means external. So I am writing down as exogenous budding. Now, in this way, there is a small outgrowth which appears first, then it further develops or it further grows, and after a certain growth, it gets detached. It finds another solid substratum, and when it gets attached to that solid substratum, it grows as a separate individual. Okay, so in this way, exogenous budding helps in asexual reproduction of the poriferans. Now, the other budding method is the internal budding or the endogenous budding method. So I will write it down as endogenous budding method. So endogenous budding, it occurs towards the paragastric cavity or the spongiocele. So if I draw the diagram with two layers, this is the paragastric cavity or spongiocele. Here we can see that there is a small bud or outgrowth developed towards the cavity. Since it is developing towards the cavity, that means inside the body, we termed it as endogenous budding. So this bud is also called as gamule. Okay, fine. So when the bud develops, this opening is called as micropyle, this particular opening. So the mass of cells is usually archaeocytes. Remember totipotent cells, so they can give rise to any other type of cells also. So when the mass of cells gets out or exit from the micropyle opening, they get attached to a solid substratum and give rise to adult individual. So in this way, gamule also helps in asexual reproduction. Okay. Now, when we talk about the sexual reproduction, then these organisms are monoecious or hermaphrodite. That means they will produce, single poriferan body will produce both male gamete as well as female gamete. Now, as you can see in this diagram, that choanocytes are differentiating and giving rise to germ cell, which is the sperm. Sperm will release into the paragastric cavity and will exit out through the osculum. When it will comes into the contact of another poriferan body through ostium or osteopore, it will enter into the mesohyoid layer or the mesenchyme layer. In this layer, there is already a female gamete or egg which is formed by archaeocyte, and when the sperm comes into the contact of the ova, it gets fertilized, and ova gets fertilized, and as a result zygote is formed. Now the sexual reproduction is in cross fertilization, internal fertilization, and the development is indirect. Now when the zygote develops, it does not directly give rise to an adult form. Instead, it gives rise to an intermediate form which is called as larva. And when this larva exit out through the osculum, it gets attached to a solid substratum, and then through metamorphosis process, this larva transforms itself into the adult young sponge individual. Okay, so this type of development in which the zygote after being development give rise to larva intermediate stage. The larva, which is different in morphological and nutritive habit, different from the adult one, such type of intermediate larval stage is being developed, and through the metamorphosis process, that particular larva develops into the adult form. Such a type of developmental process is called as indirect development, okay, through the larval stage. If it has to be directly the zygote is developing into the direct adult form, then such kind of process we would term it as direct development process, but this is not the case here. Okay, so sexual reproduction, sexes are not separate, hermaphrodite, that is eggs and sperms are produced by the same individual, and in this way the cross fertilization usually occurs, and the sperms are produced by one. Although you can see that eggs are also produced by the same body, but when the sperms get out of the osculum, they reach to different poriferan body nearby, they get internalized into the mesohylia, there they fertilize the egg and the process develops as I mentioned earlier. Okay, so the sexual reproduction occurs by indirect development. Now when we talk about the indirect development and we say that there is some larval stage, so usually there are multiple types of larva forms. On the basis of the larva, we have multiple types of larva forms, but usually there are two types of larva which we generally study, that is amphiblastula and parenchyma. So here we can see amphiblastula larva and parenchyma larva. So development is indirect, having a larval stage which is morphologically distinct from the adult. Larva is flagellated and free swimming, as we have already discussed in the third slide. Amphiblastula larva is formed in cycon type. Parenchyma or parenchyma larva is formed in leucosolenia type. Okay, so here we can see that the difference between the two larvae is the arrangement of the cells. So amphi means both. So here we can see that flagellated as well as non-flagellated cells are arranged in equal half according to this. This is the central cavity. So this is the flagellated cells arranged at one half, other at one end, and at the opposite end these are the non-flagellated cells which are arranged equally. Okay, so when it will get attached to a solid substratum, it will give rise to the adult individual. Similarly, as you can see in the parenchyma or parenchyma larva, the flagellated layer of cells forms the outermost layer, and inside the layer all the non-flagellated cells appear as a single inner cell mass. Okay, so this is the basic difference between the amphiblastula and parenchyma larva. Now, examples of porifera, these are the examples which I have given in NCERT also. Cycon or Cypha, also remember the common name. Spongilla, freshwater sponge, and bathe sponge, which is Euspongia. Okay, so these three examples, this phrase is also given, so you must remember along with the common name. Now, classification, as I have already mentioned you in the previous slide, that on the basis of the spicules also and spongy fiber or spicules arrangement, the porifera can also be categorized into different classes. So these are the three classes. Calcarea means that class which is having calcareous spicules. Okay, now examples are Cycon and Leucosolenia, which are also having radial symmetry. So you also remember radial symmetry exception. Okay, next Hexacidinalidia, which is having siliceous spicules, Euplectella and Hylonema. The diagram I am going to show you soon. Demospongia, which is including Spongilla, which is again the freshwater, and Euspongia, which is the bath sponge. Okay, now as you can see the two diagram, Euplectella, which is also called as Venus flower basket, and Hylonema, which is also called as glass rope sponge. Here we can see the ostia as well as root spicules. Now similarly, oscular sieve plate and osculum opening here also, and that's work of siliceous spicules. This is the root of which is used to establish into the substratum. So these are the further two examples. Calina, which is also called as dead man's fingers, and Cliona, which is also called as boring sponge. It usually creates pores into the shells of the mollusca. So this is also called as boring sponge. Now a short quiz. Fill in the blanks on the basis of what we have studied now. Simple one-line questions. Sponges generally lives in which habitat? Dash is the unique system of porifera. Now fill the blank related to the canal system of sponges. Ostia, then comes a middle term, which cavity, then osculum, and so on. You can see yourself, you decide the answers. I will show you the answers in the next slide. Here are some more questions. Body of Porifera is supported by endoskeletal support we have talked about. Simpler terms, common names also. So now I am going to display the answers. I hope you have figured out all correct. These are the answers. You can have a look. You can pause and you can match the answers with the fill in the blanks. Now, so this was a brief introduction about the Porifera. In the next class, in the next session, we are going to study about the Ctenophora phylum. So if you really want to get connect with us, you can scan this QR code and get registered with us and get to know more about the courses, different courses which are running out.